DNA, the DNA, the DNA, the DNA makes protein. DNA, the DNA, the DNA, the DNA, the DNA, the DNA.
Welcome to another virtual biology lecture. Today we're going to be discussing DNA replication and protein synthesis. Hope you guys enjoy and don't forget to take good notes. Today's virtual lecture's objective is to become familiar with the following assessment standards from reporting category 1 and 2 that are going to be tested in the STAR Biology EOC. At the end of the lecture, you should be able to explain the process and the importance of DNA replication, as well as the purpose and process of protein synthesis. What is it that makes us unique? Why is it that we might look similar to our family members, yet at the same time, we're completely different? Well, it's because of our genetic information. Sometimes it's even referred to as the code of life, which is found in biomolecules known as nucleic acids. Let's review briefly what this biomolecule looks like. First of all, nucleic acids are made up of essential elements referred to as CHOMP. Carbon, hydrogen, oxygen, nitrogen, and phosphorus. And remember, each nucleic acid is made up of monomers known as nucleotides. A nucleotide consists of a 5-carbon sugar molecule that's bonded to a nitrogenous base and a phosphate group. Now, cells contain 2 types of nucleic acids, DNA and RNA, and each nucleic acid is named for the sugar that it contains. For instance, DNA has deoxyribose sugar, hence the name is deoxyribonucleic acid. Now, ribose sugar is found in ribonucleic acids, which is RNA. Let's first focus on DNA, which is an extremely important nucleic acid. It carries genetic information which contains instructions for cellular activity and protein production. Now, DNA is very unique for each organism. In fact, it's so unique that crime scene investigations have made use of this to further their investigations. For years, fingerprints have been used as evidence in crime scenes because everyone's fingerprints are unique. But now, forensic scientists are actually using DNA sequencing techniques to identify individuals more precisely. So what exactly is DNA and why is yours so unique? Well, our genetic information is responsible for our unique characteristics such as cell structure and body shape, which are referred to as traits. Now, keep in mind that we're going to dedicate a whole lecture later regarding genetics, which is the study of heredity. In other words, the study of how these characteristics are passed down from parent to offspring. But for now, in today's lecture, we're going to focus primarily on a closer look at the genetic information that determines these traits. As we mentioned before, there are 2 types of nucleic acids in a cell, DNA and RNA. Let's look at DNA structure first. A molecule of DNA is made up of many nucleotides, and as we mentioned, each of these nucleotides have a phosphate group, a sugar, and a nitrogenous base. Now remember, we refer to it as the pool, the house, and the garage. Well, each nucleotide is basically the same except the nitrogenous bases. There are 4 bases in DNA, which are adenine, thymine, guanine, and cytosine. Scientists often refer to these bases by the first letters of their names. A for adenine, T for thymine, G for guanine, and C for cytosine. Now, in 1953, Watson and Crick put together all the pieces of the puzzle to determine the structure of the DNA molecule. They determined that a molecule of DNA is actually shaped like a twisted ladder. This shape is called a double helix. And as you can see in this illustration, this double helix actually consists of 2 DNA strands that are twisted around each other. The 2 strands are anti-parallel to each other, in which each strand consists of a phosphate sugar backbone, and that forms the sides of the DNA molecule. Now, the 2 strands are then joined together by the 2 bases, which always pair in the same way, following the base pairing rule, where A always pairs with T, C always pairs with G, and these actually make up the steps of the ladder. Now, the sides and the steps are twisted to form this double helix. The sequence of bases is actually what provides the genetic code to be carried in the DNA molecule. Now, it's also important to notice the sugar that's found in DNA. DNA sugar is actually deoxyribose sugar, which is why it's called DNA, deoxyribonucleic acid. Okay, so now that we know what DNA looks like, now let's take a look at RNA. Now, RNA is also made from nucleotides. However, there are some structural differences between DNA and RNA. As we mentioned before, DNA has a sugar named deoxyribose. Now, RNA, on the other hand, it contains a sugar known as ribose. And DNA consists of 4 nucleotides, A, T, G, and C, while RNA also consists of 4 nucleotides, but RNA has a nucleotide known as uracil, U, and this is actually what replaces T, which is found in DNA. Therefore, the 4 nucleotides for RNA are actually A, U, G, and C. Now, another structural difference between DNA and RNA is the way that their nucleotides are arranged. DNA is a double helix, and RNA consists of a single strand of nucleotides. There are no complementary strands involved with RNA. As stated before, DNA is extremely important to each cell in the entire organism because of the genetic code that it carries. This is why a complete set of DNA must be available for new cells produced at the end of mitosis in order to replace, repair old damaged cells, or simply for the organism to grow. Now that we have discussed the cell cycle and mitosis, you should be well aware of the importance of doubling genetic information during interphase before cell division begins in order to ensure that each new cell receives a complete copy of the genetic information contained in the parent cell. Each chromosome, which are structures consisting of chromatin, which are made of DNA, must be copied before mitosis begins in order to have 2 copies of each chromosome, which are called sister chromatids. Well, the process of copying DNA is also known as DNA replication, which consists of 3 major steps. The first step consists of the double helix to unwind, and then the 2 strands are unzipped with the aid of a special enzyme called helicase. Now, this enzyme breaks the bonds that link the nitrogen bases between the 2 strands. Step 2 begins once the original DNA molecule is unzipped, which results into 2 complementary halves, whose sequences of bases serve as a template for the synthesis of the duplicate DNA molecule. In this step, a different kind of enzyme called DNA polymerase can attach and move along the DNA strand to help add new nucleotides to the exposed complementary original strand. Now, this results in new pairs forming with the exposed nucleotides on each separate strand. If the unpaired nucleotide is A, then the only pair that it can make is T. If the unpaired nucleotide is G, then it can only pair with C. Now remember that DNA has 2 strands and they are anti-parallel, meaning one strand is upside down. When DNA is replicated, this causes the new strands to be formed at different rates. One is assembled faster than the other one, which is called the leading strand, and the slower one is called the lagging strand because it's basically assembling the new strand in sections and backwards, which causes it to go slower. Now, in step 3, this enzyme remains attached to the DNA strand until all of the DNA that was unzipped is copied. Once complete, it detaches and produces 2 DNA molecules. Each is made up of an original strand and a new strand. The 2 DNA molecules are identical to each other and to the original strand. However, it's important to note that DNA replication is not a simple process that begins at one end of the DNA molecule and finishes at the other. Rather, it occurs simultaneously at various places along the DNA molecule, which is why mistakes are made. But fortunately, enzymes check the arrangement of bases in the new strands and fixes these errors. As we have mentioned before, in eukaryotic cells, DNA is located in the nucleus. It's important to note that a molecule of DNA can be very long. In fact, the nucleus of a single human cell contains more than 1 meter of DNA. Thus, in order for the DNA to fit inside of the nucleus, it must be compacted, which is why it wraps around proteins and becomes tightly coiled into spaghetti-like structures called chromatin. If you remember correctly, this chromatin gets even more compacted when it forms condensed chromosomes. Now, we've been mentioning chromosomes, and so far we just know that chromosomes have DNA. Because there is so much of it in eukaryotic cells, it's important that the information gets categorized so that it's easier to access when needed. Think about your backpack. It isn't very useful when you're looking for a specific assignment from a class and all of your papers are just jammed into your backpack. This is why we use binders for different classes. Well, that's basically how a cell works. Your backpack would be the nucleus and your papers are DNA, which are categorized in different binders, which are chromosomes. Chromosomes are basically the categories for the genetic information that's going to be passed down from one generation to the next. Every species has a unique number of chromosomes in its cells. Humans, as we've mentioned before, have 46 chromosomes, but other organisms may have more or less as seen in this image. So why are these chromosomes, or better yet, categories, so important? Notice this chromosome is drawn with many lines or sections, which are called genes. Basically, chromosomes have many genes, which are like subcategories. Each gene can be expressed differently, which we call traits, based on the order of DNA base pairs called base sequence. So if you think about it, the dividers found in your binders are like the genes found in chromosomes. Now, it's important to notice that these chromosomes sometimes look very similar to each other, but yet they carry different information. If you notice, we tend to pair chromosomes that look the same. In other words, those that carry the same genes, we call these homologous pairs of chromosomes, which is why we tend to say that humans have 46 chromosomes in total, but 23 homologous pairs. Please note that these pairs of chromosomes may look the same because they have the same categories or genes, but they do not have the same information. Similar to how two science class binders may have the same dividers, basically same genes, but the information found inside are very different, which is how traits can be expressed differently for the same gene. Genes code for the production of proteins. Now, your body's structures are made of proteins, and as a result, those proteins help determine your traits. For example, the color of your eye is determined by a protein. Proteins are made of long chains of smaller molecules called amino acids. Let's take a closer look at the production of proteins in the cell, which is called protein synthesis. The order of base pairs along a gene forms a code that tells the cell which protein to make and what amino acids make up that specific protein. During protein synthesis, the cell reads these codes and uses that information to build a protein from different amino acids. However, you need to keep in mind that proteins are not directly made by DNA. If you remember correctly, DNA is not allowed to leave the nucleus. So in order for it to get its unique code across to the protein factory found in the cytoplasm, which are known as ribosomes, it must send a copy of the code via messenger RNA, also known as mRNA. This is actually known as the first stage of protein synthesis, called transcription, in which the simplified copy of DNA is made, known as mRNA. First, an enzyme called RNA polymerase will bind to a specific sequence on that DNA and act as a signal to start transcription. Then the two strands of DNA unwind and separate. The DNA nucleotides are now unpaired, which is where RNA polymerase starts to move along the nucleotides of the DNA strands and link complementary RNA nucleotides following the base pairing rules. Transcription will continue until RNA polymerase reaches a signal on that DNA strand that tells it to stop. Then, the RNA polymerase will detach from DNA and release the RNA molecule. For instance, let's say that this is a DNA strand with the following sequence that's going to be copied to produce a specific protein. Then, using this DNA strand as a guide, enzymes direct the synthesis of RNA. RNA nucleotides in the nucleus are assembled to form a strand of RNA. Now, as a result of base pairing, the DNA sequence above makes the RNA sequence below. Notice where the nucleotides U replace T nucleotides in RNA, which is a key indicator that transcription is occurring, not DNA replication. And now that this RNA has been synthesized, it will now be allowed to leave the nucleus into the cytoplasm as a messenger RNA, also known as mRNA. Because RNA structure is similar to DNA, it can also carry the same sequence of base pairs along a gene, in which sets of 3 bases form a 3-letter code known as a codon. Codons are actually what identify which amino acid must bind next for the specific protein. Now, there are 20 common amino acids, which can be arranged differently to make millions of different proteins. Now, during protein synthesis, the cell reads the 3-letter codes along the mRNA in the ribosome where protein production takes place. This leads us to the next stage of protein synthesis, known as translation, in which the codes found in mRNA is translated into amino acid sequences with the help of the ribosome and tRNAs. Now, let's take a closer look at what this would look like. mRNA will attach itself into the ribosome, where tRNA, also known as transfer RNA, carries amino acids to the ribosome so that they can be joined together to make proteins based on the codon sequence found in the mRNA. Now, in order to translate this code, mRNA codons must join with the correct anticodons on the tRNA. An anticodon is a set of 3 nitrogenous bases on a tRNA molecule that is complementary to the codon on an mRNA molecule. We can use codon charts such as these to decode the amino acids represented in this mRNA. For instance, the first codon here is AUG. And if we look at our codon chart, we can see that when the first base is A, second is U, and the third is G, it will code for the amino acid methionine. Notice that this other chart will also provide the same amino acid. It's just arranged differently. You will get a chance to practice with these charts in class, so make sure that you are familiar with them. Now, as we mentioned before, tRNA will deliver these amino acids to the ribosome, where they're going to be attached to other amino acids that have already been brought to the ribosome. Remember that the amino acids that will link together will be determined by the mRNA. As the tRNA continues to deliver the needed amino acids, the ribosome assembles and connects chains of amino acids to make the desired protein. Now, these chains of amino acids are called polypeptide chains, which will eventually begin to fold and form what we know as proteins. Okay, let's take a look at the overall picture here. DNA is so important to organisms that an exact complete copy has to be found in each cell in an organism. DNA basically carries the genetic information that codes for specific proteins used to express certain traits. Now, these traits are found in genes of chromosomes, which is why cells must go through DNA replication before cell division, in order to ensure that every cell gets a complete copy of that genetic information. DNA replication basically consists of the parent DNA unzipping a new complementary base pairing to be created in order to form 2 new strands. As we mentioned before, this genetic information carries genes which code for specific proteins that express certain traits. So whenever cells need to make these proteins, they must go through protein synthesis, which consists of 2 stages. Transcription, where mRNA is made from the DNA template strand, and then translation, which is where mRNA attaches at a ribosome and tRNA decodes the genetic code found in mRNA. tRNA delivers the appropriate amino acids, and a polypeptide chain begins to form, resulting into the desired protein. Okay, now that you've gone over DNA replication and protein synthesis, you should be familiar with the following assessment standards from reporting category 1 and 2. This concludes our virtual biology lecture on DNA replication and protein synthesis. I hope you all took great notes and wrote down any questions or concerns that you may have so that we can discuss in class. Well, that's it. Have a great one.
DNA, the DNA, the DNA, the DNA makes protein. DNA, the DNA, the DNA, the DNA, the DNA, the DNA, the DNA makes protein.